Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing GAT B question paper conducted in the year 2024. Please do like and share and subscribe my channel. If you want still previous year question papers of GAT B, you can go to my channel Omni Bio Entrance. In the playlist you will find GAT B, there you will find previous year question papers also. Then coming to the first question, a biochemist is pelleting down the microsomal fraction from a sample using ultracentrifuge at a speed of 20,000 rpm. What would be RCF? RCF is relative centrifugal force if the diameter of the rotor is 7 centimeters. So there is a formula to calculate RCF. It is 1.118 into 10 raised to minus 5 into R into RPM square. And radius, it is diameter divided by 2, so 7 by 2, that is 3.5. RPM is 20,000. Substitute the values and you will get it around 15,652, which is almost approximate to the first option, that is 15,680. So the first answer is the right answer. Then which of the following in which of the given centrifuge rotors the value of R minimum or radius minimum, R max, radius max, RAV, that is average radius, have the minimum deviation? So it is vertical rotor only. So answer is option 2. Then next one. mRNA, match the list items in list 1 with items in list 2. So mRNA is involved in RNA splicing. So A4 is there only in one option. Sorry. mRNA, it provides a template for translation, also can be taken into consideration, not 3. Okay. Okay, A2 can be, or A3. Okay, A3, that is provides template for translation. tRNA, it carries amino acids for translation. snRNA is involved in RNA splicing, and siRNA inhibits gene expression. So the answer here is option 1. Then next one. Which of the following cell type has the highest surface area to volume ratio? Now RBCs, they have small volume and a larger surface area. So they have high surface area to volume ratio. Not fibroblast, keratinocyte, hepatocyte. Then next one. How many grams of albumin and aspirin will be required to set a reaction between 1 millimole of albumin and 0.5 millimole of aspirin, given the molecular weight of albumin and aspirin in Dalton? So 1 Dalton is 1 gram per mole. Okay. So 1 albumin is, now multiply the number of millimoles by the molecular weight. So 1 millimole into 67,000 grams per mole. So convert this millimole to mole. So 1 into 10 raised to minus 3 mole into 67,000, that will give you 67 grams for albumin. For aspirin, the same way you have to calculate, it is 90 milligrams. Be careful with the unit. So the answer here is option 2, 67 grams and 90 milligrams. Then next one. Of the amino acids listed below, which 3 amino acids can undergo post-translational modification? So the correct answer is serine, threonine and tyrosine. Serine, threonine and tyrosine. Because they can undergo post-translational modification, that is phosphorylation, that is addition of phosphate group to their hydroxyl chains. or to their hydroxyl groups. That will be more correct. So serine, threonine and tyrosine. Then what is the common feature of the following peptides? GKWLY. Again here if you see there is GKWLY. Here also you can see GKWLY. Here also you can see GKWLY. So they are of the same amino acid composition. What is interactome? So it refers to the complete set of interaction between protein, peptides and other molecules. That is like protein-protein interaction, protein DNA, protein RNA, protein ligand interactions. Okay. Then next one. What is the final concentration of NaCl upon mixing 10 ml of 10 millimolar NaCl with 990 ml. So 990 ml of 10 millimolar NaCl. So molarity is not changing. You are diluting it. So 10 plus 990 will be 1000 ml of 10 millimolar NaCl is what you get. So what is the concentration? Convert it into, they have asked in millimolar, molar, millimolar. Millimolar is 10 millimolar. No, where 10 millimolar is present. So convert it into molar. So that will be 10 into 10 raised to minus 3 molar, that is 0.01. So the answer here is option 3. Now which of the following molecules is a peptide nucleic acid? So a peptide nucleic acid, as the name suggests, it is a synthetic, first of all it is a synthetic molecule and it mimics the structure and function of DNA or RNA but with peptide like backbone instead of the sugar phosphate backbone. Okay, so it is having a peptide like backbone instead of sugar and phosphate. So here this option 1, if you see, you can see a sugar and a phosphate backbone here and there are no peptides here. So that cannot be the answer. Here also sugar and phosphate and base. So it is not a PNA. Now this example, if you see, it has got a peptide here. This is the peptide. It is not a sugar phosphate backbone and a base is attached to it. Base is your nitrogenous bases. So that could be a PNA. So the answer here is option 3. Here it is just a CONH bond peptide but there are no bases here. So fourth one is also not the answer. Then among wheat, moong dal, rice and bajra, the one with the highest protein content is bajra, wheat, moong dal. Of course it is moong dal. Then next one. Which one of the following is the most effective strategy in delivering a gene of interest in non-proliferating terminally differentiated cells? So it is adeno-associated viral particles and it can deliver genes to, as they have mentioned in the question, non-proliferating and terminally differentiated cells. Examples include neurons, muscle cells, etc. So it is adeno-associated viral particles. Retroviral particles, they require cell division to integrate into the host genome. So it is not suitable for non-proliferating cells. Calcium chloride and lipofectamine, they are transfection methods and are effective in proliferating cells and not in non-proliferating cells. So that is also not correct. Then next one. Human adeno-associated virus is used to deliver single-stranded DNA as a vaccine that does not require multiple booster doses. Such vaccines are generally administered along with the adenovirus or a herpes virus to avoid multiple booster doses. Here both are correct. Okay, that is both A and R are correct, and R is the correct explanation of A. Then next one. In case of prokaryotes, the start codon is usually preceded by a sequence complementary to the. So this is your mRNA 5' to 3' direction. This is your start codon. It is preceded by a shined Algarno sequence, and this shined Algarno sequence, it is complementary to the 16S rRNA sequence on the 3' side. Because on the 3' side it is having an anti-shined Algarno sequence. It is to that region it will bind during translation. So the start codon is usually preceded by a sequence complementary to the 16S rRNA. Answer is option 1. Then given below are two statements. One is labeled assertion and the other is labeled as reason. In eukaryotic genes, Tata box aids in transcription. Yes, it aids in transcription initiation. So that's correct. Tata box facilitate formation of pre-initiation complex for transcription initiation. Yes, that is correct. It also serves as a binding site for Tata binding proteins, and these Tata binding protein, they will recruit other transcription factors and RNA polymerase II to the pre-initiation complex. So that explains the assertion. So both A and R are correct, and R is the correct explanation of A. So first one is the answer. Then which one of the following cell types is involved in retaining tattoo ink? It is macrophages. So when the tattoo ink is injected into the skin, the macrophages, they will engulf the ink particles. They will break down it into smaller fragments, but the ink particles, they are larger to be removed in size, to be removed by the macrophages. So they remain in the skin, and the tattoo ink will persist. So that's what is happening. Then which one of the following does not refer to secondary structure in protein? So beta sheet is a secondary structure. Twist, it is a super secondary structure. So it is not a secondary structure in itself. Alpha helix is a secondary structure. Loop also is a secondary structure. Or loop is a protein chain that connects two secondary structures. Then next one. Starting with a single cell, what will be the number of cells after n cycles of cell division, given that in each cycle every cell divides into two cells? So they have given in each cell division one cell splits into two daughter cells. So first division 2 cells, second division 4 cells, that is 2 into 2. Third division it will be 8 cells, that is 2 into 2 into 2. So after n divisions it will be 2 raised to n cells. So the answer here is option 4. Then next one. The process of nuclear envelope breakdown during prophase is not aided by which one of the following? Extension of philopodia. Yes, that's correct. Philopodia, they are finger-like projections of the cell membrane and they are involved in cell movement and adhesion. And it has nothing to do with nuclear envelope breakdown during prophase. Phosphorylation of nuclear membrane proteins, yes, this help in the disassembling of the nuclear envelope. Cytoplasmic microtubule dynamics, it is involved in the reorganization of cytoskeleton during mitosis. So third one is also correct. Nuclear lamina disassembly, yes. Lamina, it is a network of proteins and they provide structural support to the nuclear envelope. So for disassembly of the nuclear envelope, the nuclear lamina will be disassembled. Then which of the following is derived from ectoderm? It is the nerve. Muscle derived from mesoderm, bone is also derived from mesoderm, blood is also derived from mesoderm. Then regulatory B cells or Bregs are important mediators of adaptive immunity and function mainly via the secretion of. So Bregs, they are a subtype of B cells, and it is involved in suppressing excessive immune responses, such as it will suppress inflammation, inhibit activity of effector T cells, like that. So they function via secretion of interleukin 10, IL-10. Then next one. A polymerase chain reaction yield 1.2 billion copies of DNA in 30 cycles. How many cycles would be needed to obtain 300 million copies? So number of copies, you know, it is given by the formula 2 raised to n, where n is the number of cycles. So it is given 2 raised to 30 is 1.2 billion copies. So 2 raised to n, you will get 300 million copies. Now divide both the sides by 2 raised to 30. So 2 raised to n minus 30 is 1 by 4. So 2 raised to n minus 30 is 2 raised to minus 2. N minus 30 is minus 2 and n is equal to 28. So the answer here is option 4, that is 28 cycles. Then next one. Neoschisomers are restriction endonucleases with. So neoschisomers, they have identical recognition site but different cleavage site. So here if you see the example of SMA1 and XMA1, you can see they have identical recognition site. It is GGG-CCC, but they cleave at different site. So SMA1 cleaves here and XMA1 cleaves over here. So the answer here is option 1, identical recognition site but different cleavage site. Then next one. Telomerase, an RNA protein complex, adds telomeres at the end of the chromosome. Yes, it is during the DNA replication, because otherwise if the telomerase doesn't add these nucleotides at the growing ends of the chromosomes, then it can result in shortening of the chromosomes during DNA replication. So what kind of enzyme activity does it possess? It possesses the reverse transcriptase activity, or it is RNA dependent DNA polymerase. RNA dependent DNA polymerase is 3. Then next one. Which of the following statement is not true for the layers of gastrula? The lining of the digestive tract will be formed by endoderm. That's correct. The bones will be formed by mesoderm. The nerves will be formed by ectoderm. The skin will be formed by mesoderm. The skin is formed from ectoderm, not mesoderm, and they have asked for not true. So fourth is the answer. Then which of the following statement is not correct? Transcription take place in the nucleus of eukaryotic cell. Yes, mRNA formation. So it take place in the nucleus. That is correct. In prokaryotes, mRNA is not capped. Yes, it is a modification. This capping occurs only in eukaryotic cell. So that's also correct. Translation of eukaryotes take place in the nucleus. That is not correct. So in eukaryotes, what happens is transcription, it occurs in the nucleus. Then the mRNA is processed and modified in the nucleus. Then the mature mRNA is transported out of the nucleus into the cytoplasm, and the translation take place in the cytoplasm. So third is the answer. They have asked for not correct statement. So answer here is option 3. In prokaryotes, DNA replication is replicated in the cytoplasm is also correct. Then next one. Rancidity in spoiled foods is mainly due to proteolytic enzymes. Proteolytic enzymes, they break down proteins leading to spoilage. So it has nothing to do with rancidity. Photosynthetic microbes, they are algae and cyanobacteria and not involved in food spoilage. Saccharolytic microbes, they break down sugars leading to fermentation, not rancidity. So lipolytic microbes is the answer. Lipolytic microbes, they have got lipase enzymes that hydrolyze the triglycerides into fatty acid and glycerol, and this will result in bad flavors or off flavors and odors, so becoming rancid. The helical content of a protein can be directly determined using, so it is circular dichorism. It is involved in the determination of secondary structures as well as helical content. Okay, then DNA conformation is left-handed in, you all know it is Z DNA, B DNA, A DNA are right-handed. Then next one. Which of the following tissue culture approaches is most appropriate for the production of double haploid plants? Protoplast fusion, it involves fusing of protoplast from different plant species to create hybrid cells. Then embryo rescue, it is culture of embryos from which are obtained from wide crosses, or I can say interspecific crosses. So that is embryo rescue. Anther culture, yes, that is for haploid plants. Because in anther culture there are microspores in them, that is pollen grains in the anthers, and when you culture them they will develop into haploid plants which can be converted into double haploids by chromosome doubling. Then meristem culture, it is for production of virus free plants. So fourth is also not the answer. Then which one of the following is not an auxin? Indole acetic acid is auxin. It is a natural auxin. Indole butyric acid is a synthetic auxin. 2,4-dichlorophenoxyacetic acid or 2,4-D, again a synthetic auxin. 6-benzyl-aminopurine is a cytokinin. It is synthetic cytokinin. So 4 is the answer. Then which of the following is a heuristic algorithm that works faster than those driven by dynamic programming? So it is BLAST. It is a heuristic algorithm and it works faster than dynamic algorithms such as Needleman and Wunsch and Smith and Waterman's. Needleman-Wunsch is for global sequence alignment and dynamic programming algorithm. Okay. And Smith-Waterman is again a dynamic programming algorithm for local sequence alignment. And gradient descent, it is an optimization algorithm used in machine learning.